Hey, molecular genetics friends. I know we haven't talked in a while. I have not forgotten you. I've been kind of distracted, focusing on getting Lucy Koger's senior seminar up and running, and it's actually all about repeat sequences that are used for forensic DNA analysis. And so you know a little bit about that, and she'll be doing her presentation this Friday. And the next set of topics we're going to get into and kind of finish up with the semester are going to be on control of gene expression, which is really a critical and really important part of molecular genetics, kind of the nuts and bolts about how genes are being turned off and on. And developmental gene regulation is something that I'm particularly keen on. But I thought, before we jumped into that another thing we often talk about in molecular genetics is the immune system. And particularly right now with the announcement that Pfizer made at the beginning of the week that they have a vaccine that is looking to be around 90% effective at blocking the COVID-19 virus, I thought, wow, we really can't pass this opportunity up. So we're going to talk about the immune system. And we'll make sure the lights are full on. Hold on a second. Oh, that's all the light I got. All right, so we're going to talk about the immune system and how antibodies are generated and some of the different terms and things. And so this is chapter 18, more or less, depending on what kind of volume you're in of the book. And a couple of the ideas that we've got, some terms. There is a vocabulary to this biz that antigen is the thing that you make an antibody against. So in the case of a vaccine, you're injecting an antigen. And an antigen is then going to induce an immune response. And there are a number of different immune response categories. There's what's known as cell-mediated immunity, which is using killer T cells, and they recognize fragments of foreign antigen presented on target cells, and that becomes part of the immune system story. But the more critical part is something that is known as humoral or acquired immunity. And humoral or acquired immunity is what you're generating with a vaccine. And the way it works is this. You inject an antigen into a bunny rabbit or a 4-year-old screaming, and the antigen is going to interact with B cells. And we've got these specific cells that are part of the serum, and circulating what you think of as white blood cells. B cells produce antibody or antibodies. And initially, they're on the cell surface. And so, if you have an antigen, like the virus, that binds to the antibody on the cell surface, it's going to have an effect of which this cell is going to be triggered to divide more and more, and you begin developing an acquired immunity. Now, here is the thing that's really quite interesting, is that at any one point, you've got a whole range of circulating B cells. Each B cell has a unique antibody production system. And it has been calculated and sort of blue skied back in the 80s that a standard mammal, rabbit, mouse, human, can produce somewhere around 1 million different antibodies or respond to 1 million different antigens, which is pretty significant. The amount of DNA that it would require if each one of these is a separate gene is what would take up all of this random DNA that's not coding for anything. And so back in the 80s, when people were doing Koch curve analysis and beginning to look at the complexity of genomes and seeing that the human genome actually had a lot of repetitive DNA, one of the notions that before this was really fleshed out that was posited, that was put forward, is that potentially a bulk of the human genome is devoted to producing antibodies. Well, it's not that way at all. And it took quite a while, and David Baltimore may have won the Nobel Prize for it, to determine exactly how this works. So it's a very interesting story. And what you got then is these circulating B cells, and they've got antibody on their cell surface. And they have the ability to bind antigen. Now, when they bind antigen, that causes a signal to be sent out that causes that cell to begin dividing. And so you have a system going on here that the tighter the antibody binds to the antigen, the more rapidly it divides, and you begin to produce an immune response. You begin to generate antibodies against the antigen. So let's take a look first at, when we talk about antibody, what we're talking about. And if we take a single B cell and look at the antibody that is being produced, one moment, please. I'm just thinking about things. All right. This is what it looks like. It's a fairly large protein complex made up of two subunits. One subunit is called the heavy chain, and we have two identical heavy chains, and the other subunit is called the light chain. There are two identical light chains. So you have the heavy chain here, and then the light chain here. So two heavies and two lights. Okay. Now, within the amino acid sequence here, you can begin to look at the amino acid sequence, and it turns out that this portion of the heavy chain is constant. This is known as the constant region. And we'll see in a moment that there is a portion here that is not quite constant. This is the antibody class determining region. We'll get to that in just a moment. So we've got the constant region, and there's a constant region in the light chain. And just for you biochem nerds, it is the amino terminus is up here on both of these, and the carboxy terminus is down here. And we then have a region, and let me see if I have a different colored marker that I can use. Up here, that is known as the variable region. So, we've got the constant region and the variable region. Now, when we look at antibodies in general, the antibodies that cells are producing, the constant regions are all identical. The variable regions are different between individual B cells. All right, we had to take a pause for a moment, but we're back. So, we've got the variable region and the constant region. So it's the variable region that is within these two parts, the heavy chain and the light chain, is, as the name implies, different between B cells. That's where the variation comes. And if the antibody protein shape can bind the antigen, then you have an immune response. All right, so just another vocabulary word. On the antigen, let me see if my blue marker works. On the antigen, we've got the actual spot where the antibody is binding. So that antibody binding site is known as an epitope. And one B cell can be producing an antibody that binds to a specific epitope on an antigen. So when you inject an antigen into a rabbit or a person, you begin to bind to B cell antibodies that have been produced in the B cell in multiple spots. And so you could have an antibody that binds to one portion of the virus and an antibody that binds to another portion of the virus. And so these are separate kind of binding events. And these are known as epitopes. And this is what's referred to as a polyclonal antibody, because you've got a whole range of different kinds of antibodies being produced now in the body. So the next step that happens is something that is known as class switching. So, as we were saying here, the initial antibody that is present in a B cell is actually membrane bound. And so it will look something like this, where you've got the heavy chain and light chain, and it's absolutely transmembrane protein, and it's present on the cell surface. So now when an antigen binds, it's going to trigger this cell to go into division. Now, once an immune response starts, after a couple of weeks, the B cell is going to keep the same variable region, but it actually will change this specific spot. And this is known as class switching. And we end up having an immunoglobulin that is membrane bound, IgM, can begin to class switch. And what we start working towards is a form of the immunoglobulin that is now soluble, the immunoglobulin G. There are a variety of different classes that we'll get to in just a moment. And these are responsible for either circulating in the blood, or they're found in secretions, things like IgA. IgE is another class that's part of the allergic response. IgM is the initial type, which is involved in the establishment of the antibodies and things. And so we've got a variety of different kinds of types here. Now, the question then becomes, how do we generate antibody diversity? And it's a good question. Glad you asked. And it turns out that when you look at the regions of the genome that contain the heavy chain and the light chain, they have a similar kind of structure, and it's a little bit like picking one from column A and one from column D. It looks like this. There is a phenomenon that occurs in B cells that is really quite rare, and it's something known as somatic recombination. Now, let's unpack this for a moment. We have recombination that occurs in gametes. Remember, during meiosis, you have crossing over in females. But once you have, the fusion and you produce a fetus, the embryo, the little child that begins running around, a small bunny rabbit, those somatic cells, their DNA is pretty much, immutable. It doesn't undergo recombination. But in the B cells, it does. And that's the trick behind this. And so let's look at the light chains first. The light chain gene is arranged like this. So we've got a series of what are known as the V genes, And then we've got the C genes, the variable regions and the constant regions. And what we're going to do is have a range of different recombination events such that we've got a variable region in which we can have, and we're just looking to see, not that you need to worry about the numbers exactly, but what are the numbers? All right. There are approximately, in humans, 300 V regions. And then there are a set of J sequences that are associated with then the constant. So you have a series of JC repeats. And what you're going to do in order to generate a functioning light chain is you undergo a somatic recombination in which the genome is actually rearranged, and you'll pick one of the V regions, say V region 173. And you then undergo recombination to fuse it with one of the JC regions. And as it turns out, the way it works is there is control sequences out here, that is the promoter, that are only going to fire once recombination has occurred. So once recombination occurs, you have actually built this gene. And so you've got a possibility of 300 different variables and at least 4 or 5 J regions. And so the number of possible combinations is really quite high. And they're going to undergo this recombination event. Now, the heavy chain is assembled in a similar way in which you have the variable regions. With the heavy chain, we have an additional spot. Instead of just having the J and constant, we're going to have the variable and then something known as the diversity or D region, and then the J constant region. So we have VJ recombination in light chain and VDJ recombination in the heavy chain. And in terms of the heavy chain, in humans, there's again around 300 V regions, something around 20 or so D regions, and then there, I think, are about 6 J regions. And then we've got the constant regions. Now, this recombination event is going to initially involve just VDJ recombination in the heavy chain. Once the immune response takes off, essentially once a B cell binds to an antigen and yells, "Hooray, I'm it, somebody loves me," it actually then goes through yet another round in which the constant chain begins to switch. And this is what we know as class switching. So what's going to happen is, and you know, this is a standard kind of tool to make antibodies. You can do it with a vaccine. I've done it with bunny rabbits, where we have a protein that we isolate from an organism, flies, bacteria, whatever. And in terms of practical, actual production of antibodies, you take the antigen, and it can be denatured, it can actually be in native shape, whatever. You typically will mix it with something that is going to act as an irritant. And I always feel bad for the bunny rabbit because, you know, you're going to actually whoop up on them pretty hard. It is something that is known as adjuvant. Adjuvant is a material, typically kind of a mineral oil-based material, that's going to cause the antigen to be released slowly. And you have something that's called complete antigen, or complete adjuvant, which actually has some killed bacteria that are going to just elicit a big immune response in general. Pretty nasty. I think with vaccines that they give to people, you don't typically have anything too aggressive in the adjuvant, because otherwise you'd end up running a pretty serious fever and feel pretty lousy for a couple of days. As it is with a vaccine, you probably are going to have potentially a mild fever and have some soreness there because you are eliciting an immune response. So you have the adjuvant and the antigen, and then you inject your bunny rabbit. And typically you do a subcutaneous injection. You grab the bunny rabbit and then using a syringe, slide it into underneath the skin, and you inject half a mil or so of this mineral oil protein goo. And then you rub it in there and you say, "Good bunny," and you put him back in his cage. So you start on day 1. And the general rule is, in terms of the progression, we have injection on day 1. And you may have heard this from the flu vaccine and things, is that you don't have immune response for a few days. Or at least a sufficient immune response so you can actually fight off the virus. So you can get the vaccine today, and you get exposed to a virus within the next 2 weeks, you might very well still be sensitive to that virus, because it's going to take a while. And so generally, we inject on day 1, and then 2 weeks later, day 14, we're going to do a booster. Now, for humans, sometimes when you're a little baby and you had vaccines, you might get a booster shot. Depends on the vaccine and what kind of immune response you're trying to generate. Sometimes you don't. One shot will be enough. A booster will definitely help. And that just kind of kicks things along. So what's happening is, on day 1, we've injected the antigen. It binds to the antibodies that are on the cell surface of the B cells. Those B cells that bind, that have undergone the appropriate somatic recombination, they begin to replicate. Now, just as a little point, somatic recombination occurred in utero. You've got the B cells that you're going to have. And so you have a wide range of different B cells with different antibody types, but what you got is what you got. All right, so you're selecting for those that are binding. And the booster shot is going to further maintain this. And one of the things that also starts to happen is you get somatic mutation, that there are systems in play in B cells that allow them to actually tweak or mutate the VDJ regions of both the variable, the light chain and the heavy chain. And if you can bind a little bit better, then you divide that much faster. And so it is a little bit of like a microevolution, in that your immune system is fine-tuning itself to generate antibodies that bind the very best. So you give a booster shot. And then probably 7 days later, so day 21, you'll do the first bleed. Because the initial immune response is going to be IgM. Around here is when we start to get class switching, and we switch over to IgG. So IgM is membrane bound, IgG is soluble. It's actually being secreted into the blood. And now we're going to take blood, spin out the red blood cells to generate the serum, the crude serum. That has got the antibody in it. Now, there are all kinds of technologies for purifying antibody. We'll talk about them in just a moment. But the serum can absolutely work. And what you do is you do some kind of antibody antigen test. An ELISA, which is an enzyme-linked immunosorbent assay, and there are a lot of ways you could do it. The easiest way to visualize it is you take the antigen, you put it in the bottom of the plate, let it dry down, you add antibody from the blood, you rinse it. The antibody that binds, you can then add a secondary antibody that's going to bind that first antibody, and that secondary antibody will have some kind of enzyme or a radioactive tag or a fluorescent tag, and you measure the amount of signal you get that way. And so this is a standard thing that we do all the time around here, is using an ELISA system. You can also do a protein gel blot, where you run a gel with the antigen in it. You blot that onto a sheet of nitrocellulose, soak that in the antibody, and you look to see what the concentration is. One of the first things you would do when you're trying this is you do a series of dilutions of the serum. And in that way, you can begin to identify what's known as the titer, which is a measurement of the antibody concentration. All right. We will pick up some more antibody stuff on Thursday, and I'll check in with you then.